Hi guys, how you doing? So obviously I'm away today. Mr. Calford and I are representing Eastdale at a conference for grade 9 math teachers. So that's exciting. But we are going to carry on in this lesson talking about sex-linked traits. So we're going to just finish up that note we had from yesterday, and then I'm going to give you the duration of the period to work on whatever it is you have to work on. So let's take a quick look here. That's the one I want. All right. So yesterday we started talking about the fact that not every trait is independent, because we have, yes, 46 chromosomes, and 22 of those chromosomes are, or 22 of those pairs, so 44 of those chromosomes are what we call autosomal chromosomes. And the autosomal chromosomes are chromosomes 1 through 22, and they are the ones that everybody has 2 copies of, with very few exceptions. So some people who have an extra chromosome or something like that, but almost everybody has 2 copies of chromosome 1 through 22. But the remaining 2 chromosomes are what we call the sex chromosomes. And there are 2 different sex chromosomes, the X and the Y. And we said the X chromosome has about 800 genes, lots of coding genes about lots of different things. But the Y chromosome only has a small little handful, only about 70. And the Y chromosome contains genes, mainly one gene called SRY, which activate male sex characteristics. To be clear, that doesn't mean that all the genes for being male are on the Y chromosome. That's not the case at all. Most of the genes for being male are spread out among all the chromosomes. But the kind of master control gene that activates those other traits more fully is found on the Y chromosome. So generally speaking, people who are biologically female have a copy of the X chromosome that they get from their mother, and a copy of the X chromosome they get from their father. Whereas people who are biologically male still get an X chromosome from the mother, because that's all a mother has to give, but will have a Y chromosome from their father, their biological father. So from there, we talked about the idea that knowing this about chromosomes, not every pair of genes can be randomly assorted. You can't always just do a Punnett square, because it's possible that genes will be found on the same chromosome. And if they're found on the same chromosome, they will likely split together. So in this case, where we had one chromosome that had the recessive copy of A and the dominant copy of B, those are likely to move together because they are on the same chromosome. Unless crossing over happens, an egg cell or a sperm cell will likely have those 2 genes together. And likewise, these 2 would tend to move together. And crossing over would have to happen in this section to get a case where you could end up with the homozygous recessive or the homozygous dominant. So those are going to be less common. All right. And we left off talking about diseases. I want you to keep in mind that you will be doing a disease project in this unit, kind of like you did in the previous unit as well, about genetic diseases. So do pay attention here. Make sure you get this down. So we said there's basically 3 types of diseases. There are what we call autosomal diseases. And unsurprisingly, those are the diseases that occur in the autosomal chromosomes, chromosomes 1 through 22. And these are caused by a mutation of some sort, a change in the DNA sequence. And depending on the nature of that change, it may be the case that simply having one bad copy of, I shouldn't say bad copy, of just having one mutated copy is enough to cause that disease to manifest itself. But it may require 2. And we said, for example, if you had a dominant disease, in that case, if you were homozygous dominant or heterozygous, you would be diseased, and you would have to be homozygous recessive to be normal. On the other hand, if the gene is recessive, you would have to be homozygous dominant. You would have to be homozygous recessive to be diseased, and either the homozygous dominant or the heterozygous individual would be normal. Now, mind you, we would say that this person would be considered a carrier because even though they themselves don't have the disease, they would be carrying a copy of the gene. From here, I want to talk about something related more specifically to the lesson today about sex-linked chromosomes, is you can have sex-linked diseases. Now, these are also caused by a mutation in a gene, but in this case, instead of being caused by a mutation on a gene in one of the autosomal chromosomes, these are caused by a mutation in a gene on one of the sex chromosomes. And just like the autosomal ones, they may be dominant or they may be recessive. Now, let's talk about this. For a recessive disease, that was found on the X chromosome, we could have XN for normal or X lowercase n for disease. And you'll note that we've included the X here. We've done that to demonstrate specifically that this is a disease found on the X chromosome. So we could say X uppercase N means an individual who has the normal case, and X lowercase n is an individual who has the disease phenotype. And in this case, if you're female, because remember, females have, people who are biologically female have two X chromosomes. Well, they could either be XN, uppercase, the homozygous dominant, in which case they will be normal, non-diseased. They could be heterozygous with one dominant and one recessive allele, in which case they'll be normal, but we would consider them a carrier. Or they could be homozygous again, but this time with the recessive, in which case they would be diseased. So for a female, person who is biologically female, to get a disease of this nature, they have to have two recessive copies of the disease. They would have had to get a recessive copy of the allele from their mother, their biological mother, and a recessive copy of the allele from their biological father. On the other hand, a male only has one X chromosome. They have a Y chromosome, and then they have an X, and that X can either be the normal gene or it can be the recessive gene. But they don't have two copies. If they have the normal gene, the normal allele rather, they will have the normal phenotype. But if they have the recessive allele, they'll be diseased. There's no such thing as a male who is a carrier of a recessive trait if it's on the X chromosome, because they only have one X chromosome. We can look at an example of this. Hemophilia. Hemophilia is a recessive trait found on the X chromosome. It's typified by an inability for blood to clot properly. So somebody who's a hemophiliac will tend to bleed and continue bleeding. This is kind of a famous example of a sex-linked trait because, again, the gene that controls blood clotting is found on the X chromosome, and there's a diseased allele of this gene that causes blood to clot improperly, to not really clot. And for people who have hemophilia, a small cut can be very dangerous because they will continue to bleed without proper clotting factors. So let's look at this case where we say that a hemophiliac mother and a non-hemophiliac father reproduce. And given that they reproduce, what percentage of their female children will have hemophilia? Before we do the Punnett square, why don't you pause this and kind of think about that just quickly. What percentage of their female children do you think will have the disease? Again, keeping in mind that, like the example above, this is a disease that's found on the x-axis. So in this case, we could use hemophilia as an example. Normal, and the disease case would be hemophilia. You don't have to write that here. But anyway, let's give it a try. So pause this video and try it without me. See if you can do a Punnett square and tell me what percentage of the children will have hemophilia. Okay, well, let's do this, shall we? So we know that the mother is a hemophiliac, and since this is a recessive disease, that she will have to be homozygous recessive. So she will have to have a diseased copy of the X chromosome and a diseased copy of the allele on the X chromosome. And the father, on the other hand, we know is not a hemophiliac. And keep in mind that for him to be normal, he will have to have the normal gene, and that's the only copy he will have because his other gene will just be a Y chromosome. He doesn't even have an X chromosome to give. So he must have the normal case. So this is the mother and this is the father. So now let's see if we can make a Punnett square. All right, well, let's do that. Sorry, I'm just trying to be neat here. It makes me feel a little bit crazed when I'm not neat. Okay, and let's put the maternal genes up here. So let's take the mother's alleles and put them here. And a mother is either going to donate this diseased allele or that diseased allele. So the mother is always donating a diseased allele. And the father, on the other hand, he is always donating either a Y chromosome or a normal allele. So either a Y chromosome or an X chromosome that has the normal allele, just like that. And with that in mind, we can look at their offspring, and we can say this one would have a Y chromosome and an X with the disease. This one would have a Y chromosome and an X with the diseased. This would have a normal X and a diseased X. And this would have a normal X and a diseased X. So let's look at the genotypic frequencies. We have YX little n being 2 out of 4, which is 50%. And we have X dominant and X recessive being 2 out of 4, which is also 50%. Okay, good. After we look at the genotypes, we should look at the phenotypes. Well, as it happens, this phenotype is male because they have the Y chromosome, and they are diseased because the only copy of the X chromosome they have has the diseased allele. So diseased males, diseased and male is 2 out of 4, which is 50% of the offspring. And this allele is female because they have 2 X chromosomes, biologically female, and will be normal, though a carrier. So normal female is again 2 out of 4, which is again 50%. And so to answer the question, the original question is what percentage of their female children will have hemophilia? We can say none of the females in the F1 generation will have hemophilia. All of the males will, as it happens, though that isn't the question. There we go. That is good. We could also say all of the females will be carriers for hemophilia. If you wanted to, you could pause right now and figure out that if these, the women of this family were to go out and marry, or not marry or whatever, but reproduce with men who did not have hemophilia, what percentage of their children would have hemophilia? You could look at a next generation, but we're not going to do that here. But we can simply say that despite the fact that all of the females are normal, they are all carriers for hemophilia, which is noteworthy. Sorry, just got a text. All right. So that's two types of disorders. We can have diseases that are autosomal in nature. These are mutations that are found on an autosomal chromosome. We can have sex-linked diseases. These are diseases that are caused by mutation found on a sex chromosome, the X chromosome or the Y chromosome. And the third example we will look at is what we call chromosomal disorders. And these are caused by having an extra or a missing chromosome. You're going to do a little bit of research on these, so I don't want to go into it too much, but the most well-known example, and the most common, is called Trisomy 21. And Trisomy 21 means that there is a third copy of the 21st chromosome. And this is also called, it's also known by another name, Down syndrome. And Down syndrome, or trisomy 21, is typified by developmental delays and often intellectual impairment. So having an extra chromosome affects the development of a fetus and the development of somebody into the adult organism. So trisomy 21 is not the only chromosomal disorder, and we're going to do some work with that tomorrow when I'm back in class. But it's just one example. And like I said, you're going to do a disease project. So I'm not going to get into too many examples here. But note that these can also relate to the sex chromosomes. For example, there are individuals who are triple X, have 3 copies of the X chromosome. There's a condition called Kleinfelder syndrome. Which again, I'm not going to get into in too much detail here because some of you may choose to study it, but just be aware that there are examples of chromosomal disorders that involve the sex chromosomes as well, and these obviously have impacts on the secondary sex characteristics, but not just on the secondary sex characteristics, in other ways as well. So that's the end of this lesson. For the duration of the period today, you can get caught up on anything you don't have done for me. All right, make good use of your time, and I will see all of you all tomorrow. Auf Wiedersehen.